Good afternoon, good evening, or good morning, wherever you are. My name is Dr. Samuel Ayodele Egeye. I have the privilege of presenting some session of this course on molecular docking. I'm a senior lecturer at the University of the Western Cape, and I lead a research group on computational pharmacology and chemoinformatics. Um, our focus is computational drug design using bioinformatics, chemoinformatics, machine learning, and data science to find druggable targets and compounds um, that can be designed into drug candidates for infectious and non-infectious diseases. So, in this course, I will be taking you through um, molecular docking, but there will be some sort of a prerequisite a conversation we're going to have prior to that in terms of looking at biological databases. Chemical databases will also look at molecular visualization and manipulation using different software packages. Then we'll now delve into molecular docking. Okay, so I'm going to start by looking at biological databases and chemical databases in this particular session. So we'll start with biological databases. So just for the avoidance of doubt, uh, kind of define what databases are. So usually they are electronic systems um, where you have an organized collection of structured information or data, and the electronic system is usually your computer system. And these days, the structure of databases has significantly improved from what we used to be, um, where you know have information in different fields, in different servers, now being pulled together into a central server, and that central server, once a query comes through the interface by the user, is able to go to that central servers, which links to all the servers where the informations are stored and give an output to the user. And so significantly, the structure around the connection between the individual servers and the central server has improved. We used to have relational database system pioneered by Oracle, but now we have what we call it graph-based um, system that is a bit more intuitive, and the connection and the network is a bit more complex than you have in relational databases. And the visualization of the data rather that you see once you make a query is significantly better these days. Okay, so you can get more information if you really want to, if you're interested in looking at the structure of databases. But if we now move to biological database, if we want to define that based on the definition we've given to databases, um, then we can say that it's an organized collection of structured life science information or data that are collected from scientific experiments, published literatures, some high throughput experiments, sometimes from industry and academic research. So even sometimes some computer simulation that have been done by other people. Okay, so all this data are put together and organized and structured in such a way that the user can query and get what they want. And sometimes the database goes ahead to not only give you what you want, interpret the results that you get from such a query. Okay, and the reason why, let me put it this way, the emergence of biological data is actually a response to large amount of data, biological data that have been generated especially from DNA sequencing. Okay, just let me check something before I move on here to be sure that. Okay. All right. So, what kind of information do we have in these databases? Excuse me. So, I'll just quickly remind you of the central dogma of molecular biology, where we know that the DNA or the gene. The DNA is transcribed into the RNA, and the RNA is translated into proteins, and you have proteins that are involved in biochemical pathways. And of course, this eventually leads to the phenotype of the things that we observe in the lab. Okay, so most research in this field try to draw a correlation between or amongst the phenotypic observations and these other um, path that or processes that take place within the cell, referring to the genomic, the transcriptomics, the proteomics, and metabolomics processes that, of course, we are known now are responsible for some of the phenotypes that we observe in some of our experiments. Okay, and so if we have a lot of information on this and all these processes, we are able to uh, give better explanation to the biological phenotypes that we see. All right. So genome, in terms of specific information in this genomic, which we're going to be like focus on a bit, will be your gene ID, the gene sequences, the gene expression levels, the function of the gene, the structure of the gene, and the rest of them, and the proteomic information specifically looking at the protein, the name of the protein, the gene that is responsible for that expressed protein, the function of that protein, the location of that protein, and how the protein interacts with other protein in the biochemical pathway or signaling pathway within the cell, the expression level, the domains, and the rest of them. So this typically just shows you the kind of information that are available within biological database. It's by no means exhaustive, and you will see more as you get into your hands-on experience activities that you will be doing in each of this after I finish this series of conversation with you. So if we want to say what are the types of databases that are available, we can generally classify into sequence databases, structured databases, and let's call the others and put the rest of the databases on under others. So according to this author, so sequence databases, it makes sense because you have your nucleic acid sequences, your protein sequences, and there are databases that store this information. Um, they might not be limited to that, but they are basically set out to store information related to the sequences of nucleic acid, and like SwissProt set up to store information related to protein sequences. So you also have structured databases that naturally now, um, after the translation, able to uh, scientists have resolved the structure of some of these macromolecules and proteins, and they are able to now deposit the structures of those proteins in a particular database, like your protein data bank, you know. And the other refers to like functional or what we call a secondary databases that take information from these kind of infer functions from either from the sequence on the structure or analyze the structure to give us better information. So you now have, for example, I think I have something on the next slide that talks about that. So you have nucleotide databases like your GenBank, protein sequence database like your UniProt, your structure database like your PDB, protein data bank, and you have what we call the domain um, or motif databases that actually now look at the structures themselves and try to define, excuse me, domains within that particular structure. So you have your gene, like I said, gene bank, nucleotide sequences, you have your protein sequences, you have your protein structure databases, and you have some other databases that also explain uh, part of that particular protein in terms of looking at specific domains and motifs. So domain is a particular is essentially a conserved part of the protein that can perform its function or move independent of the other parts. So this is another list that I just made in terms of biological databases. Um, Once again, it's by no means exhaustive. There's quite a large number of databases. So for a specific research area or interest, I can guarantee that you will find a database that has curated and annotated information from different sources and put in that particular database. Okay, just to mention at this point, um, if you do an experiment and you're confident of the data that you have, you can actually deposit your data in some of these databases. There's a process in which um, the owners or the people that maintain these databases, when they get such information from researchers, they actually take them through quality control checks, and in terms of really checking the experiment behind that particular data, now try to now explain what the data is in terms of the process of annotating that data, what are the different parts of that data, what do they mean, and then putting an identifier on that particular different aspect of that particular data, and now curate them, trying to put them in specific themes, and in order to make it easy to query, so that people, if they're looking for gene related to, for example, to viruses, you should be able to see any information or research data from that under that particular uh, theme, as it were, or particular structure that they have, or architecture that they've built for that particular database. Okay, so why do we need them? Why is it important? Why don't we just go to Google and type in your thing and say, okay, give me the gene for virus, give me the protein for virus, okay, give me the biochemicals that are related to virus, to this protein that you have in that particular virus that is, expressed by this particular gene. And, just talking about it is, it's already making me tired, you know. But a biological database it's a one-stop shop that gives you access to a variety of biological relevant data, you know. You can have data ranging from literature, scientific literature, the genome sequence of a particular organism, the gene identifier themselves, and the description of that gene, and the variation in those particular genes, um, alternate splicing, transcripts, and the rest of them, the structure of protein that result from that gene, biochemical pathways, the chemicals that can modulate that pathway, the disease that result from a particular gene or particular variation. So, the list is beyond this circle that I have here, you know. But what's your biological database? What they bring to the fore in terms of value is that they provide the infrastructure, they provide the architecture for you to be able to have to prepare this database, to manage this database, this particular information, and to mine information from this particular collection of information. So they make it very easy for us to go in there. For example, if you look at this figure, you have this network built by this spider web, in which case basically we're saying that the genome sequence is connected to the taxonomy of that organism, phylogeny, to connected to the biochemical, connected to the disease that is observed from that or a particular phenotype in that particular organism. Okay, so, and this network, there, it's very difficult for you to just collect data and build this kind of connection. It's very, very difficult. So the computer system does that for you, based on the architecture behind the specific database that you are querying. Now, because these systems are stored within the computer system, this database rather, and the information they are in are stored within the computer system. We can do a lot with it in terms of simulation. Doing test mining is a part of your machine learning protocol can be implemented, and sorts of other sort of simulations that we can do. Molecular docking itself is a simulation, and you need to engage with the database, get information that you will now use for that simulation. Okay, so it makes it easy for us. We're not just, if the structure of your protein is on the paper, your scientific paper that you printed out, you know, It's not good. It's not good for it to anybody in terms of simulation. But now you have a way where we have transferred that sequence or that structure into a computer readable form. Then we can take that computer readable form and using some other algorithm and begin to engage with that particular structure. Now, the first biological database was GenBank, which is a collection of all available protein and sequences um, submitted by NIH and NCBI. And the collection of this database actually gave birth to the project of the Human Genome Project, which, because of the advancement in DNA sequences, we are able to sequence the whole genome of a human being. But that was the easier part. The sequencing was the easier part. The most difficult part, the more difficult part was the making sense of the data that came out of the sequence. After doing all the cleaning and calling of the bases and the rest of them. The question now is you sit with a series of ACTGs in different combinations. What does it mean? Where is the gene? Where is the gene for that particular biochemical process? Where is the gene for that particular phenotype? Where does it start from? Where does it end? Where's the exon? Where the introns part of that gene? Where are the regulatory part? Where's the promoter region and the rest of them? You need to now account for that, and that is when bioinformatics actually came in to begin to annotate that particular data. And by annotating them, you can now bring them into a database such that if you say find the gene responsible for angiotensin converting enzyme in humans, you can actually go to that particular chromosome and the loci for that particular gene, and you'll see the starting position and the end position, the promoter region, and the rest for that particular gene. Okay, because somebody has taken time to sit down. Not somebody, a group of people. I remember a couple of my supervisors were involved in that project a long time ago. In the South African National Bioinformatics Institute, and yeah, to put that project together. Of course, beyond that, we've done quite a number of things in terms of looking at variations, because having established the genome for humans, we've been able to now come specifically to Africa and say, okay, what does the genome of an African look like? What does the genome of a particular ethnic group in Africa, what does it look like? And that is another information that determines the kind of phenotype we see, why these people are able to get this susceptible to this disease, and this other set of people are not. Or these people can use this drug, medicine, these other people cannot use this medicine. And we're able to, make sense of that also. So you have a database that goes beyond just the gene bank, looking at genetic variations and the rest of them. So all these databases helps you to, based on the research question that you have, you can always find a starting information that can help you build your hypothesis. Okay. So specific examples of biological databases include all these that I'm trying to list here. You have fungus or Saccharomyces databases, viruses have databases also. I particularly use Plasmodb because the focus of my research was on malaria. Plasmodb is part of the VEU PathDB, basically for pathogen, eukaryotic pathogen. And I also use this a lot, Protein Data Bank, which is going to be our focus in this session. UniProt is another, we're going to be looking at this also. It's a comprehensive resource for protein sequences and function. It tries to give free access to sequences of protein and annotate those proteins in terms of the function they perform. And I think it also gives information about the subcellular location of such of those proteins. So It's very important for us to, um, let me put it this way, that if you to have an understanding of the function of your protein, because we in medicine, different types of biotechnology, expressing, is particularly recombinant DNA technology and the rest of them, drug design, and different aspects of biology. We really want to have an insight. We really want to have an insight into the function of a particular protein. So, UniProt provides that platform, and is very up to date, and a very comprehensive information about proteins. Um, it also helps to not only collect but also interpret sometimes some of this information that are present within that particular database, and it saves researchers countless hours of collecting information yourself and trying to make sense out of it and everything. You know, and it's links to almost over 150 databases. So if you take a protein and you look at the function, you can actually now begin to say, okay, what are the domains with this particular protein? It links you to that particular database that stores information about the domains of protein, which gene actually express this protein. It takes a link and it takes you to the database where the gene for that particular protein is stored, and you can actually begin to look at that gene within the gene browser and the rest of it. Okay, so it's quite a functional, and I found it very useful, especially when it comes to drug design, structure-based drug designs particularly. Okay, so perhaps this is central for anybody that is doing structure-based drug design. And a central technique to structure-based drug design is molecular docking, which is the focus of our discourse here. Excuse me. Excuse me, I need to put my phone on silent. Okay. So I was saying protein data bank is a three-dimensional structural database of large biomolecule, not just protein, nucleic acid, and some complex systems that are formed either from the interaction of two protein or a protein and nucleic acid, and the rest of them. Okay. But the kind of information you have here are the three-dimensional structures that have been resolved for a particular protein. We know protein usually exists as your tertiary structures. So from your primary polypeptide chain, they form your secondary structure, which is your either a beta sheet or an alpha helix that now folds into your tertiary 3D structures. And of course, you can also have two proteins, 3D proteins, interacting to form what we call a quaternary structure. Okay. So yeah. But the whole idea here is that having access to this 3D structure, for example, if we talk about our focus, which is molecular docking, we want to be able to identify um, the active sites, or what we call it binding sites, for a particular protein. So we need to be able to, for us to do that kind of simulation, we need that information within the computer system. And this information is basically people who have taken time to use X-ray crystallography or NMR, nucleic magnetic resonance spectroscopy, to resolve the structures of this protein. So what I mean by resolve is basically to account for imagine you have this long chain that is folded into this. You are able to account for if the first part of this chain is alanine 1. The question is, where is alanine 1 in this final structure of the protein? So by accounting for each of this in the final structure, you can build the three-dimensional structure. So you have a coordinate for each of these amino acid that's part of that polypeptide chain in space, where they are. And that is a starting point for us when you want to do your simulation in terms of molecular docking or any other simulation that you are interested in. All right, so I'm going to do a live demo in terms of engaging. I'm going to look at UniProt. I'm also going to look at Protein Data Bank. Then you're going to have an hands-on exercise that you're going to try on your own, and I would suggest that you take notes as you try them. And your on-site and online tutors will also be available to assist you. I'm also available to assist if there are things that you think the online or on-site tutors cannot handle. Okay. All right. So I'll get around to that in a minute.